Today I would like to explain to you the concept of the synapsis of chromosomes during meiosis. Recall that meiosis is the process of creating 4 unique cells from a single original cell, where each new cell created has half of the original genetic information. Meiosis occurs in order to create the unique daughter cells for the purpose of procreation. So, to explain this concept of synapsis between two chromosomes, I have 2 examples I will show you. So for the first example, imagine an individual who has 1 original cell with 2 homologous chromosomes here. Homologous chromosomes mean that each chromosome contains the same genes that carry genetic information. Also notice that each chromosome has this dot somewhere in the middle called a centromere. This will help us to understand the orientation of the chromosomes as we work through these examples. So our first individual has 1 chromosome with the genes M, L, I, period, J, K, H, G, and another chromosome with the genes G, H, I, period, J, K, L, M. As you can see, the chromosomes are in fact homologous because they each contain the same genes. So for synapsis to occur, the chromosomes must be oriented so that all the genes match up. Now, I'll draw a line connecting the chromosomes where the same gene exists between each chromosome, as well as draw in the centromeres between the J's and I's. So, the way we can see what possible chromatids, which are identical halves of a divided chromosome, are created is to follow along each line I've drawn. I like to imagine that a race car travels along each track, and the genes that the car passes over are the order in which the newly created chromatid is organized. So, I'll show you on the top track first. So the race car travels over a M, L, I, and then the centromere, and then a J, K, H, and a G. So now things are going to get a little more complicated. Now I need to explain the concept of crossing over. This occurs when parts of the chromosome swap places in order to create new combinations of genetic material. Let's imagine we have crossing over between the J and K genes in these chromosomes. Well, as our race car travels along the second track, we will first cross the M gene, then L, I, centromere, J, and then we cross over to the other track because of the crossing over, and we next encounter K, L, and M again. So on the next track, we pass M, L, K, J, centromere, I, H, and G. The last track, we have to start from the other side to avoid producing the same chromatid twice. So first we pass G, then H, I, a centromere, J, K, H, and G again. So for the chromatid to be viable, it needs to contain all of the original genes from the original chromosomes. As you can see, only the first and third chromatid contain the M, L, I, J, K, H, and G genes and would be considered viable. For our second example, let's consider a second individual with the chromosomes M, L, I, J, K, H, G, with a centromere between I and J, and a second chromosome J, H, K, J, centromere, I, L, M. You might have noticed that this is actually the same order of genes, just reversed. So we can line these 2 chromosomes up evenly to understand the synapsis that would occur between them. In this example, we will still encounter crossing over between the J and K genes. So the first track, we will pass M, L, I, the centromere, J, K, H, and G. The second track, we will pass M, L, I, centromere, J, cross tracks, K, H, and G again. On the third track, we will pass M, L, I, centromere, J, cross tracks, K, H, and G. And unsurprisingly, the last track produces the exact same chromatid yet again. Because they all contain the original genes from starting chromatids, they are all viable. So we can now evaluate the fitness or ability to produce offspring between both of these individuals. The first individual will produce viable chromatids 2 out of 4 times, or 50%, while the second individual always produces viable chromatids 100% of the time. Therefore, we can conclude that the second individual is twice as fit as the first individual and will be much more efficient at producing offspring. I hope this helped you understand the concept of synapsis between chromosomes during meiosis. Thanks.